For the merfish, okay. For the merfish, the principle behind the merfish is very similar to the one behind the sackfish. So we have multiple hybridization rounds, and the good point of this technology is that they performed this computational error correction for the imperfect hybridization. So I suppose that in this case we have a more accurate identification of the transcripts in the during the experiment. So it's less time consuming than the first version of the sackfish. And also it has been used in combination with other technology by the Brain Initiative Self-Consensus Network for building an atlas for the mouse brain. So just a more couple of slides. So here it is to summarize a little bit the world of the spatial transcriptomics technologies, because the spatial transcriptomics technologies will be even more adopted in the future. And if you want a review on these technologies, there is this review which is a very brief review. They distinguish more categories for these technologies, but in general, all of them can be reconducted to the two families that we distinguished. And we are seeing and evolving on these technologies during the years, where for the spot-based family, we saw that the spatial transcriptomics, we had more cells for just one spot, and now we are reaching a resolution to have multiple spots for just one cell. And this can be achieved with the stereoseq, for example. And while for the molecule-based, we have these sackfish and sackfish plus, where in the first case, we have just hundreds of molecules that could be detected, and now we have thousands of them. And also there will be, I think in the next few years, and they are still emerging, multiple spatial technologies able to detect multiple modalities for each cell with the spatial information with them. For example, we had already a 10x technology able to detect transcripts together with proteins, and some others able to detect also some other epigenomics factors together with the transcripts. This is a very brief overview of the tools that we have for the general spatial analysis. For example, in R, we have the SF and SP packages. They are very general tools for the spatial analysis, and some people are starting to use them in combination with other bioconductor tools for the analysis of the single cell spatial data. There is this Giotto package, which is specific for spatial transcriptomics analysis. They provide several BigNet and examples with several technologies, not just spot-based, not just the 10x Visium, for example, but also some molecule-based technologies. While the CURAT that probably everyone knows, it's a more general framework for the single cell analysis, and they showed how to analyze spatial technologies, but their BigNet is focused mostly on the spot-based. They have examples with the next Visium and with the SlideSeq data. Outside R, in Python, we have this SquidPy package, which is specific to spatial transcriptomics and has been developed by the same developers of the ScanPy tool. And I think that could be some interoperability between the Python and the R packages in Bioconductor in the future. So to conclude, I want just to give an overview of the Bioconductor project for the ones that maybe don't know. This project is aimed to provide statistical analysis and comprehension of the genomics data. It includes more than 2,000 packages developed together by the core team of Bioconductor, and also from external researcher from the Bioconductor team. They have almost 1 million downloads every year for the packages, and also more than around 50,000 citation in the PubMed Central. It is also a very open community. So if you want to join the community, you can use the Slack channel. And I invite you to do that also, because you can very easily participate to the discussion, contribute to the development of packages, and also just asking for help to the community. And in most of the cases, someone will answer your question. You have also the support website and the general website for Bioconductor. So at this point, I will leave the stage to Helena, that she will talk about the Bioconductor infrastructure, and so at the end we could have some discussions if you have any questions. Thank you.
Okay, thanks, Dario. Second. There. Okay. So before diving into the spatial realm, I would just wanted to start a bit with what we had at the time in Bioconductor before spatial came about. So about 6 years ago, Bioconductor put out the Summarize Experiment class, and shortly after the single cell experiment class. And this has become the go-to structure for storing data from any single cell experiment. And just to quickly summarize, this class contains assays, which can be primary or transformed data, such as counts or some log transformed normalized counts or other expression-like values. And here, every row is a feature and every cell a column. The features needn't be genes, they can also be proteins. We have row and column data that can store metadata associated with every sample and observation. And then specifically in a single cell experiment, we can store reduced dimensions such as PCA, Tisen, UMAP, or other cell embeddings. We can store experiment-wide metadata. This can be anything, any sort of object that we would also like to store. And finally, fairly recently, there is the Alt-XP slot to accommodate data for the same samples, but different features. For example, in CiteSeq, where we measure both proteins and transcripts. Over the years, there has been lots of development around this class, and the perhaps greatest demonstrations of this is the OSCA book, Orchestrating Single Cell Analysis with Bioconductor. This is really a great resource, a comprehensive online book, both for teaching, but also self-learning, and just giving an overview of all the infrastructure available in Bioconductor. And this comprises theoretical background on technologies, the single cell experiment class, pre-processing, downstream analysis steps. And because this is so great, we want to leverage a lot of this, because many of these methods are in fact applicable to spatial data. So we wanted to build on the single cell experiment class in order to make use of everything that's there. And for that, we built the spatial experiment class to inherit from the single cell experiment class. So we automatically get the assays, the row, the call data, and the reduced dimensions. In addition, we can have spatial specific metadata. For example, the position of a spot on the chip, or a logical indicator whether an observation maps to the tissue or not. However, these are not that special, so we just store them inside of the call data. Secondly, we have spatial chords. So these are our spatial coordinates, and they behave analogous to the reduced dimensions. So these are a numeric matrix. Now, importantly, if we store multiple samples in such an object, these spatial coordinates will overlap, or they can overlap because every chip is almost the same. And for that, we have a protected sample ID slot in the call data, meaning that it cannot be removed and it cannot be arbitrarily replaced. And the purpose of that is simply to track which coordinates belong to which sample. And most importantly, we have the image data, and I will talk about that in a lot more detail on the next slide. And this stores all image-related metadata. What does that look like? So the way it currently is after many iterations is a capital D data frame, where every row is a unique image entry that has a sample ID and image ID associated with it. For example, we could have multiple images for the same sample in different resolutions. We have a scale factor that tells us how to rescale our spatial coordinates to overlap with the image, because that depends on the resolution. And the data slot, which is the image itself. And for that, we provide the spatial image class, which in itself is virtual, meaning it is empty. And from that, we have three subclasses. First of all, the loaded spatial image class that is really realized into memory. And the image is here stored as a raster object, which is simply a matrix where every row and column is a pixel, and the entries specify a coloring. We have a stored spatial image. So this can be sitting on a computer somewhere, and it is only loaded into R upon request. And thirdly, the remote spatial image class, where our image can be hosted under some URL. And again, it is only retrieved upon request. And for this image data, we implemented a couple methods. So first of all, get, add, and remove image for accession, addition, and removal of spatial images. And secondly, the image raster and image source successors to retrieve the data stored inside of a spatial image. So for a stored spatial image or a URL, that would be the location of the image. And for a loaded spatial image, we could retrieve its raster object. And here's just a quick example of what that looks like in action. So we could specify some URL, add that image to our spatial experiment, extract the raster object, and then simply plot that with base R. Okay, so in spatial experiments, what can happen is that this image that we get is not aligned with our spatial coordinates. And for that, we implemented two methods to fix that. The rotate and mirror image functions. So first of all, here's just an example of counterclockwise and clockwise rotation, which is done via the rotate image class, which takes a spatial experiment and a degree value. And secondly, the mirror image function, which just flips the image or mirrors the image horizontally or vertically. And again, this is required because there's programmatically no easy way to figure this out. So if a user were to notice that the image and the data don't match, they would have to fix this once, and then everything would be okay. Okay, next, in terms of molecule-based data, as Dario already mentioned, we can have multiple observations associated with every feature and observation. So we get a lot of XY coordinates of our molecules, and then the gene and the cell that this molecule has been assigned to. So there's no one-to-one mapping. And for that, Aaron Lund suggested the bumpy matrix class, which he also developed, for holding non-scalar objects in a matrix. And from such a data frame, it's really like a one-line command to construct this. And to more easily visualize this, we can think of a skyline where every row is a feature, every column is an observation, and these entries are stacks of values. This can be a bigger matrix, of course. In our case, it's XY coordinates, and every position can have a different number of values or a different height. And within the SPE class, we have a designated molecules accessor for the molecules assay to store the molecule coordinates. In terms of object construction at the time, the biggest data was coming from Visium, and 10x Cell Ranger pipeline had a very consistent output structure, so that's good news. We knew where the data was coming from. However, reading this in is quite cumbersome. So we have to read in our scale factors, our spatial data, our counts, images, and then somehow construct this object. And because we didn't want the user to have to go through all that pain, we implemented the re10x Visium constructor that simply requires specifying the directories where this data is stored, and then constructs a spatial experiment from it. Of course, for other technologies, the data might not look like this. In fact, it looks different every time. So we can also construct an object similar to a summarized single-cell experiment using the spatial experiment constructor. This, in the simplest form, requires really nothing. We could just provide some counts. We could pass some spatial coordinates, so a numeric matrix of X and Y coordinates, or include these in the call data and tell the constructor which columns in the call data correspond to spatial coordinates. Similarly, the image data can be passed to the constructor. However, oftentimes it's easier to add images downstream because for most of the analysis, we might not even require having the images. And lastly, we also support coercion from the single-cell experiment class and vice versa. Now, in terms of data, currently, since the last release, there is the 10x Visium data package, which is a collection of the 10x Genomics Visium data sets available at the time. So these are shown here. There are 13 data sets from 23 different samples across human and mouse and 13 tissues. And for those familiar with the Experiment Hub, it's a few lines of code to retrieve such data to use in development for teaching or a bigger analysis project or benchmarks, etc. There's also the ST example data package by Lucas Weber, and the single-cell multimodal package also contains a seq-fish single-cell RNA-seq multimodal data set. And all of these are in spatial experiment format, the spatial data sets. Okay, so in terms of visualization, Lucas and I developed the GigaSpavis package. This is designed to work with the SPE class. It supports both spot-based visualization and molecule-based visualization. An example of the latter is the bottom right plot. And there's very flexible options for coloring, highlighting spots, and overlaying data with the image, which is from experience not straightforward. So this requires rescaling the spatial coordinates, fixing the plotting device to really have the spots overlay with the image, etc. So it's really useful to have wrappers to do all that. Also to mention, there is the IC package, the Interactive Summarized Experiment Explorer, which is a Shiny-based graphical user interface for interactive visualization. And this allows for deployment of custom panels. So anyone can go ahead and implement such a panel. And luckily, the SPE inherits from the SCE, which inherits from the summarized experiment. So we can use IC to visualize data. And with that, there is lots of room for extensions specific to ST data. First and foremost, probably just to plot spatial plots. And I think there's already people working on this, or I'm pretty sure I know that that's true. Also to mention, there is the SpatialLibD package, which provides a very complex Visium data set of 12 samples, 3 biological samples, each with 2 replicates at 2 different cut heights of the brain, which I think is maybe one of the most complex data sets out there. And they also provide a more general Shiny tool for visualization that is also based on Plotly, so it's interactive and allows for exporting visualizations and results. And lastly, many, or I hope some have heard of Spotlight, which is a method that came out last year for deconvoluting spots using a single cell reference. And this uses NMF regression to predict for every spot the proportion of cell types that it is made up of, because at this time a spot might contain 15 different cells or something like that. Now, when this first came out, unfortunately, it was only on GitHub and very strict in working with Seurat. So one would have to run Seurat to run Spotlight, and it wasn't straightforward how to do it any other way. Luckily, I'm now in Barcelona, and I could convince Mark to rewrite the entire package. And it's been submitted to BioC top of the year, and now works also with a summarized experiment class, or simply with a matrix. Okay, putting everything together, Lucas has initialized the orchestrating spatially resolved transcriptomic analysis OSTA book. This is still very early days. This screenshot that you see here is mostly a snapshot of the table of contents, but many steps are still in progress. And hopefully this will include a theoretical introduction of different technologies and the SPE class, pre-processing such as segmentation, and analysis workflow. And again, many of these things are from single cell analysis. And then there are some things specific to spatial data, such as deconvolution. And finally, there will be workflows for selecting spatially variable features, spatial clustering, etc. And I'm sure that with time, as the community contributes more methods, this book will grow. To summarize, we currently have a class for storing spatial transcriptomics data, GigaSpavis for visualization, IC and SpatialLibD for interactive exploration. There are a couple of data packages and the OSTA book, which is aimed at putting everything together. However, there's lots of open issues or opportunity for future work. First of all, what happens if we have multiple different segmentations for our data, or multiple binnings when our data is subcellular resolution? So this appears in, for example, StereoSeq and HD Visium. Then we arrive at different samples, but the same set of features. And currently, the alt-XP is the reverse. So there's no straightforward way of storing multiple different segmentations or binnings. Secondly, it's often come up in discussions how to store geometric data, such as polygon vertices or graphs for our samples. Of course, there's workarounds for both of these issues, but it would be nice to come up with an elegant and streamlined way of dealing with this. There's room for ST-specific IC modules, which I hope are also coming soon. And first and foremost, there's very little to none BioC-based analysis package for spatial data. So I think currently most people that I know write their own pipelines, kind of wrapping around methods. And it would be nice to see the community contribute a bit more here now that we have some infrastructure. And lastly, as Dario mentioned in the beginning, we would like to be interoperable with Python infrastructure that exists, mainly SquidPy, which is fairly comprehensive. Currently, this is somewhat possible by exporting data from R and re-importing it into Python, but that's not very safe, maybe. Okay, and for both Dario and I'd like to thank a couple of people. Lucas, who is our joint author here on the spatial experiment, GigaSpavis, the ST example data package, and also started the OSTA book. Aaron, which with his expertise in everything, has been of great support regarding development and implementation ideas. There has been lots of input from the BioC community through Slack and conference discussions. We're being supervised and controlled by Stephanie and Davide, who are having regular meetings with us and have been really of great support in everything. Sheila, Leo, and Brenda have been very useful in early discussions regarding the class and providing continuous feedback when actually working with the class of how things were going. And also Leo and Brenda put in a lot of time and effort to keep the SpatialLibD up to date with frequent changes and working with the spatial experiment class. And I'd also like to thank the Hein Lab here in Barcelona, where I've had really great access to lots of spatial data to hopefully be able to get out some future developments by having access to very complex data. And with that, I'm through, and we'd be happy, Dario and I both, to take questions.
Yeah, excellent. Thank you so much for this fantastic overview. Super informative, Dario and Helena. I forgot to record right from the start, so we are missing a little bit the first 5 minutes. I'm sorry about that, Dario, but the good thing is Dario and Helena agreed to also share the slides in addition to this recording that we're going to publish on the Bioconductor YouTube channel. Before I'm opening here room for questions, I know Robert wanted to say a couple of words. So Robert, if you just would like to speak up.
Great, thanks Ludwig, and thanks very much Dario and Helena. I just wanted to make sure everybody knows that Ludwig is trying to work with as broad a community as possible to start to bring together some talks, really more focused on the analytics and the infrastructure that will underlie them, but informed by the kinds of questions that we ultimately want to answer. And so hopefully folks here will give them their email address if they want to be on for more of the talks. We have a number of people already lined up, and hopefully we'll post that list and then get input from folks here as well as to what to do. I think as our speakers outlined, there's an awful lot of work to do, a bunch of, I think, really open questions and opportunities, and it's going to be, I think, really challenging for us to succeed if we don't find ways to collaborate and then sort of pick off important problems in that. And amongst the ones that I think are really key are ones of trying to understand how to get the real value from both the spatial data and the sort of omics level data. I'm more in the camp thinking that we need to be able to go back and forth between them and think about looking at sort of clustering of cells on the image side for morphology and location, and then being interested more in similar sorts of things for cell type identification on the omics side. And then as Jeff Moffat sort of informed us when we were looking at this a few days ago, you know, the other challenge is that a real experiment is 50 terabytes of images or thereabouts and likely to grow. And how do we find ways to interact in a sort of principled way with something that's that scale? It's all nice to pull up raster images and show them and flip them, and we'll ultimately, I think, do that for subsets of anything we see as ways to investigate. But the reality is that the images are really going to be big, and we don't have currently the same level of analytics within or expertise within Bioconductor for how to deal with those. And then the last thing I wanted to say is, as may be apparent, we're planning to put some significant effort into that here at Harvard over the next few years. And I just wanted to make sure the folks that are thinking about transitions in career know to check in with Ludwig or myself. If you're thinking about changing jobs, we'd certainly be happy to talk to you about what we're doing. And then turn it over to questions for our speakers.
Excellent. Thank you so much, Robert. Along these lines, I'm just following maybe up quickly. Robert said in the beginning, this is a seminar that is going to happen weekly that Helena and Dario kicked off today. We have a number of folks that will talking in the following weeks. I think the next one that is really interesting and open to the public is here leveraging the cell ontology to classify unseen cell types, and then a couple of other things going over to visualization and analysis of multiplexed imaging data, which touches a little bit more on the imaging component that Robert also pointed out. So if you are interested in sharing the work with us that you are doing in this space, please feel free to reach out to me. Other than that, Robert touched on the image aspect, and maybe if I start off the questions to Helena and Dario, can you summarize maybe a little bit the challenges that are currently there with the images? The solutions that are there seem to be working very well already for raster image. What is the question, as Robert pointed out, when these images are getting bigger? Do we need to change to particular image formats? Helena, you also touched on this extremely interesting point. How do we go over to Shiny apps? We have some of the IC developers here, Charlotte and Federico, I saw. Of course, the right next question, as you said, would be, can we make maybe a panel that holds such an image and put some information on top of these images? Have we explored the challenges there yet, especially when these images are getting bigger?
Helena, do you want to answer that? Because at the moment, we are handling just low resolution images, because mostly with the Visium data, we designed this image data structure, which is able to load this data, but there are just a few megabytes. In general, the problem is that when working with these images, you need, so if you have bigger images, you need more memory, right? More RAM memory. So that could be a little bit tricky. I am not sure how to I think that the raster image could load higher resolution images, but I don't know how many are we able to load at the same time, for example. And so that could be a little bit of a problem when we have, I don't know, 10, 20 big resolution, with big and high resolution images. But for sure we can test that, and also we can try to come up with something new. And about the IC module, maybe maybe Federico can say something more about this, because I'm not such an expert on how to I think that we can do it, but the problem could be the same, because I don't know how much the Shiny app could be able to handle high resolution images, and I think that theoretically it should be possible, but the problem could be the same. That do we have enough memory to do that, and all these kind of questions are still open.
Yeah, I don't know, Federico or Charlotte, whether you want to say something, whether there are already any developments. If not, these would be very interesting developments where I would likely also get involved with. So I don't know whether there's already something in the works there, or whether you're planning on doing some work there.
Not that I'm aware, at least. So I would be super fond of seeing that happen, but say as of now, I don't have any experience directly on spatial transcriptomics data from collaborators, so where I can have an internal push or external push to do it. But if there's any effort that where for example, you might need some guidance on how to develop these kind of panels, because say you don't want to need the experience and say the expertise or wizardry to dig into the custom panel creation, then I'm more than happy to say at least give it a listen and see what is the plan and what is potentially feasible. Because as of now, I mean, we might be as busy and as committed as we want, but we all have 24 hours a day, even you, Ludwig, and Robert, and Helena, and Dario. So that is probably the main bottleneck, probably. But say if we kind of manage to to bring together people that have different set of expertise to speed that things up, that is probably the best outcome that we can get. And once something works in a manner that we can showcase, then it's quite easy to have it as a kind of a demo-like website, say Shiny app that we can probably have to get more opinionated input. And it's easier to trigger some feedback from someone when they have something in front of their eyes that is already working, and that would be, say, the next step. But the working prototype could be feasible.
Mm-hmm. Mm-hmm. Yeah, no, I would be super interested to follow up on this. I think, like as Helena pointed out, in SpatialLibD, these folks kind of like started up to put a prototype for this together, like having an image and then maybe selecting some features or some annotations in the call data onto this image. And I think generalizing this idea a little bit, like in something like IC, would certainly be great. Robert also pointed out in the chat a tool that already goes a little bit into that direction from Niels Gillenborg here at HMS. It's the VTest tool, and they also kind of like revisit this panel-based approach of like sample and metadata annotation and feature expression to interact with each other. So would be great to see what we can maybe like use from this VTest tool. Yeah,
there was also the VTESAR tool also, which is now in the pipeline for the Bioconductor contribution submission. So I checked it. I think that was submitted a few days ago. So it was one of the top submitted ones.
Interesting. I didn't know that. Yeah, yes,
so I mean, there's no point in reinventing hot water, and say whoever is actually further ahead, they feel free to get in touch.
Sounds good. There has been a question from Jenny in the chat on the image operations that are currently there. Helena mentioned the need to use this if you notice that the image and the omics data that you have, or at least the coordinates, does not match. I experienced this myself as well. It's very useful. And the question from Jenny here is, what does that mean, and how do you arrive at such a disaligned state, I think is the question.
I mean, what we mean by it doesn't match is simply like if you have a moon shape spots on your screen, if you plot them, and your image is a moon, but it's rotated, and you plot them on top of each other, you would immediately see by eye that it doesn't match, but it would match if you rotate it. But for the computer, I guess it would be very hard to, or it would be more complicated to kind of detect that. Like Ludwig sent a data set the other week where that was the case. And it simply happens because the image on the microscope is measured a certain way, but then maybe someone accidentally rotates it, or I don't even know, like in the lab how this happens, but then it just comes out 90-degree shifted or rotated or something. So it's not a big deal, but it's easier to fix it in R, I think, than trying to figure it out by its own. And just maybe to change a bit the discussion away from the images, regarding size, the biggest challenge that I'm facing, and maybe you have this with Murfish, is that all of the BioC infrastructure, even principal component analysis, everything wants a matrix, but the data comes in a table, and just converting that table to a matrix takes an hour. Like, I mean, I have a table that has 15 billion rows of pixel level data of an entire embryo. And I'm trying now to rewrite so much code to work with that table before I convert it actually to a single experiment or a spatial experiment, because doing that right from the start would be a huge waste of time. So that's the issue with this, with the molecule-based or subcellular resolution of an entire embryo or entire brain, is just that it's so big that it's currently really difficult to deal with that, I think.
Maybe I can toss a comment in here. So first of all, Helena, really great overview here, and Dario as well. Thanks for going in. My name's Jeff. I was one of the co-inventors of Murfish. So super excited to see more people coming in this field and helping to solve the back-end computation sides that we're facing. One of the things that I want to put in your mind is that, yes, the data size is only going to get worse, much worse, rapidly. But don't feel that you, in working with this, so as you're building your tools, think about scalability, but don't feel that you're so stuck with the formats that we, as the people developing the technologies, are giving to you. If there's something where you say, look, this should have been stored in HDF5, this should have been stored in some other way, upstream, we're writing these pipelines actively, and we can easily meet you halfway on this. So if there are places where we need to think about performant file formats and developing those specifically for these type data, or probably more likely co-opting things that have already been developed and just making those the routine use, I think that is something that we should think about. So if you are finding that you have these bottlenecks because you're trying to convert one format into another, and that format wasn't particularly well suited for the scale of data, like a CSV file or something like this, this is where I think a conversation like this can be very productive. So be empowered to say, we should have changed this upstream. What we're giving to you is not what you wanted to get. Does that make sense, what I'm trying to convey?
I think Dario and Helena, you're muted.
Yeah, I think that's a great point. Just changing the point of view of the problem. So that's what you're suggesting. And yes, so we have definitely think about this, and also discuss with others to have even more ideas as some brainstorming on this. But right now, I don't know where to start from. So that's the
I think,
for me.
Yeah, I think it can't be generally said. For the molecule-based data, I mean, yeah, it's a table, that's fine. In StereoSeq, at least we still have spots, so there's fixed observations. But if we just have molecules, every coordinate could be unique in principle, right? So you can't just make it into a matrix when you write out the file. It has to be this table. And then you do segmentation, then perhaps you get a matrix. But in the beginning, yeah, even for the StereoSeq, it's more, I think, the issue that the data, the way it is, makes sense, because if you put it into a matrix, there's 99% of just zeros. But it's just tricky because, for example, if I just want to run a PCA, I need a matrix. I can't run the PCA on this long format table. But then for other things, data table is very fast. So it's actually nice. I timed it, and actually working on the table is much quicker than working on the matrix in many cases. So it's not straightforward. And maybe also what Robert mentioned is that sometimes, we have to think about chunking the tissue and processing it in chunks instead of the whole embryo. I've also tried that, at least for development, to just cut out a slice and work on that. Same for visualization. If I try to plot the image, and there's 15 billion spots or something, I can't even see it with my eyes, right? So I have to do a chunk of it to even visualize it. And I think these are things that are really currently everyone's working on them on their own, but we don't know. Yeah,
I think you're right, Helena. I'm sorry, Davide here. I just thought I would jump in, but I think I don't know if there's an issue of scale here, right? Because I guess subcellular resolution is great to look within a cell where transcripts are co-located, but then if you're looking at the whole embryo level, you probably just need a summary at the cell level, right? So I guess working with two different levels of summarization, or I guess multiple levels of summarizations of the data, could be helpful.
Yeah, I completely agree. I would toss in here. So in the image world, this is well captured with the pyramidal representation, right? You're absolutely right. If you have a petapixel, you're not going to visualize that on your nice monitor. So you've already pre-cached lower resolution versions of the data, and then on the fly, you're loading only as much as you can actually perceive at that time. And as you're zooming in, then you're loading just those pixels that you can resolve. I think that style format is likely going to be important in the visualization tools, both for the density of RNAs and for properties like the boundaries of cells. If you do a watershed algorithm on an image that has 100 nanometer pixels, you will have 100 nanometer resolution boundaries to these cells, and just storing that, plotting that can be massively problematic. And so, of course, you don't need all of that resolution depending on the scale at which you're looking. So I think as you're thinking about these data formats, that it's probably good to think about pyramidal style representations for multiple of these types of metadata for the purposes of visualization.
No, I think that sounds great. All the discussion here, I think we really need to follow up on a couple of these aspects on the image side. We're a little bit out of time, but maybe as a last question from my side, not really a question, but maybe more like a comment or another aspect to think about these structural challenges that maybe the molecule data poses on the data structures that we have so far, which are, as Helena put out, are mainly rectangular and well suited for the spot-based data. But I think like when we have like molecule-based data, we have this data frame where we kind of like have a molecule being associated with some spatial coordinates, and that's about it. And I think until here, Helena pointed it out, we have this innovative solution from Aaron Lunn, this bumpy matrix construct, which kind of like makes the segmentation by stacking up these segments in the third these molecules in the third dimension of our matrix. And I think I'm just really interested in how this plays out downstream. So I just started to like put this data together, and as Dario and Helena said, the methods are just coming out, are emerging very, very slowly. But I'd be interested in whether that's really the way of going forward, or whether we need something completely else for the molecule-based data. So these are just some thoughts from my side.
And maybe just to add that I think at least the issue that I'm having when I'm working with this, I feel like we have a little bit of infrastructure, but there's just things missing, right? So every time I want to do something, I have to implement it myself. But in principle, for example, if we stored in the raw data, just the coordinates that we measure without a segmentation, just for every molecule, the X and Y coordinates that we get, someone could write a simple wrapper where, okay, if I give it this segmentation, that's the counts I get. If I give it another segmentation, that's the count I get. So just methods like this would be very useful for lots of people. And I think it's fairly independent of how we store the data. I think it will show with time how easy then it is to use. For example, the bumpy matrix, it looks really weird. And at the beginning, I didn't find it that useful. But in the end, actually, if you want to get the counts, you just have to count the rows in every entry. So it's actually very simple. It just takes some time for people to put out tools to work with it. And yeah, hopefully that will come in the next years like it did with single cell.
Excellent. Then thank you very much. Thank you, Helena and Dario, for this fantastic overview, and everyone else for this very interesting discussion. And with that, we are maybe closing for the day and hope to see some of you guys soon for some more follow-up and additional seminars. Thank you very much.
Thank you, everyone.
Thank you, everyone.
Thank you. Bye.
Bye.